So the plants are using that sunlight energy to create a situation to allow for lots of ATP production and another molecule that's called NADPH. Those are really what it's going for. And this quote by a Nobel Prize winner is pretty much summing up in a very elegant way what's going on in terms of what they're doing with sunlight energy. Photons is the idea of light energy. What drives life is thus a little electric current kept up by the sunshine. All the complexities of intermediary metabolism are but lacework around this basic fact. So a very elegant way to say that the light reaction process, first of all, that's a primary productivity idea where plants and other organisms that do photosynthesis are creating, so the higher plants, cyanobacteria, algae, they're doing very similar mechanisms of light reactions. And if it wasn't for them, we wouldn't be here because it also is producing oxygen, which surprisingly, I'm not going to talk a whole lot about when we focus in on the light reactions, because from the plant's point of view, the oxygen generation isn't such a big deal. For us, it's critical. We wouldn't have evolved without that, but their goal is not to create oxygen for us. It is to get the chemicals that they need to stay sustained and run the rest of their physiology, get their sugar production made, grow, reproduce. So here's some interesting quotes on oxygen, which we're very lucky that they can do that and produce that. But if you're wondering, we consume about 130,000 liters per year of oxygen in a rainforest. When all those plants are doing photosynthesis, they give off a lot of oxygen, but a desert juniper, not so much. So it really does depend where you are, how active they are in terms of photosynthetic processes. Desert plants in general have a slower metabolism, they grow slower. That's one of the trade-offs that they have for being in such a warm, consistently hot, drought-prone area. But other than these little interesting little factoids, I'm not going to really focus on oxygen because that's not why the plant is doing photosynthesis. So this idea that they can absorb light energy is pretty amazing. Thinking about light, it's radiant energy. It's part of the electromagnetic spectrum. We tend to focus on this range of wavelengths from 400 to 700 nanometers. So nanometers is the way we measure the different wavelengths. And when you think about sunlight energy coming to Earth, it really behaves weirdly, like continuous waves and discrete particles, depending on what it's doing. So as it's traveling through space, it behaves as a continuous wave of electromagnetic energy. Once it interacts with particles, substances like chlorophyll in a leaf, then it starts to behave as discrete particles of energy. And so if you want to know more about the physics of light, I'm not the person to talk to. A physics professor could get into way more about this. This is often how they depict it when they talk about it in a physics class, where you have these wavelengths going on, where you have through, like I said, as it's traveling through space, you'll have that wave property of light. And so if you haven't thought about that before, this is the basic idea of how you measure a wavelength. And so that can be really long wavelengths, really short wavelengths. And then you often want to know the frequency as well as the wavelength, and they are related. So frequency is the italicized V there, and that's equal to C divided by lambda. And the C is the speed of light. Lambda is your wavelength event. So they are inversely related to each other. And so here on the next slide, you can see that inverse proportional or inverse relationship where you have wavelengths, really small wavelengths give you very high frequencies, okay? And just the opposite at the other end, you have really big wavelengths, and then you have very low frequency. So 400 to 700 is what we refer to as the visible spectrum. That's very human centric because that's saying what we see with our retina and our eyes. It's 400 to 700. Other organisms can see beyond that, below 400 and above 700. We're limited to this range. When you pass sunlight or light through a prism, a glass prism, and it breaks into these different colors, those colors respond to different or correlate to different wavelengths. So when, let's see, this week in lab, you're going to do the light reactions of photosynthesis. You're going to actually manipulate the ability of the plant you're working with to do photosynthesis, either cause it to speed up or slow down, given the different treatments you're going to surround it by. You're going to be working with spinach. And you'll see we're going to play around with different quality of light or colors of light, which actually will be different wavelengths of light. And you'll see how they respond in terms of how quickly they can do photosynthesis when you provide like only red light or only green light, what will happen. So as you pass that light through a glass prism, you have the idea of different colors, red, orange, yellow, green, blue, indigo, violet. So I do want you to know that, and I know that that's kind of a basic idea in physics, but we need to think about this idea. So red, orange, yellow, green, blue, and sometimes they leave out the indigo, so I usually put it in small I, and violet. The way I remember this, R-O-Y, Roy, is a man's name. It's actually was my father's name, so it was very, very easy for me to remember that. And the way I remember green, blue, indigo, violet, that stands for Green Bay is victorious, because our household went with the Packers, not the Lions. So Green Bay Packers, Green Bay is victorious. So please think about that. Think about what those colors represent in terms of general wavelength ideas, where red is the larger end down at 700. And as you work your way through that acronym, you get down into the smaller wavelengths. Okay. All right. So plants will care about certain wavelengths more than others. And that all has to do with their chlorophyll. So their chlorophyll evolved and adapted to kind of only pay attention to certain wavelengths. That's the certain wavelengths they will maximally absorb and are really good at absorbing it. Other wavelengths, not so much. So chlorophyll is picky that way. So when light is interacting with chlorophyll, it will act as a stream of particles, which we refer to as photons of light energy. And if you talk about the energy, then you say it's a quantum. If you want to know the equation, it's H is max. Do I have that? Yeah. Planck's constant. And so quantum energy, E sub Q, that's quantum energy, is equal to HC divided by lambda. Remember, C divided by lambda, I defined in a previous slide, that's equal to the frequency. So you can simplify that if you want to. EQ is equal to H times the frequency. And H is the Planck's constant. I'm not going to ask you to do any calculations, but if you're interested in thinking about quantum energy with these different wavelengths, you'll be able to see that different wavelengths will definitely provide different quantum energy. So when plants are doing photosynthesis, the only way they can get this to happen successfully is they have to absorb that light energy. And like I said, that's the chlorophyll pigment that's doing that job. And they have to allow for this absorption. The goal here really is to get an electron in the chlorophyll molecule excited. That's the trick. And so you have to imagine that chlorophyll pigment absorbing that light energy, one of its electrons gets really excited, and we say that it gets boosted or elevated to a higher energy level. And they often say that's the excited state of the electron. So this is going back to like Chem 111 or Chem 112, getting into these ideas of excitation of electrons. And then once you have that happening, that'll creates a situation where that electron could get passed off now to an acceptor molecule, which is what we'll talk about. Okay, so this chlorophyll is getting excited. One of the electrons is getting excited. It really does depend which wavelength of light the chlorophyll is getting exposed to. This is a kind of a, the black line is showing like when sunlight is striking, you get this kind of input here, the irradiance. And so looking at chlorophyll, if you zoom into just the green line down here. So when the sunlight reaches the plants and their chlorophyll, you see two absorption peaks. And they've matched it up down here to the quality of light between 400 and 700, that stretch of wavelength of light. And so if you look carefully, you can see that one of the peaks gets you thinking about red light. The other peak, you drop down, that's more like blue light. So what this is saying is chlorophyll is adapted to maximally absorb red and blue light. That's the quality, those are the qualities of light that it evolved to capture and be able to really work with. So when you see peaks like that, that's saying, that's optimal absorption. And so the chlorophyll pigment is really zooming in on red and blue. That's really the wavelengths of light that it's maximally absorbing, which is good because now those red and blue qualities of light are going to excite the electron in the chlorophyll pigment. All right, so let's say that's happened. Let's say the chlorophyll is getting hit with red and blue energy and that electron is getting excited. There's a few things that can happen. It could just get excited and then come back down into its unexcited state. And then when it's coming back down, that extra energy is given off as heat. So as the electron gets excited, but then nothing happens, like it doesn't get donated off anywhere, then it'll just come back down into its lowest excited state. And as it's doing that, it releases heat. That's not really what the plant's going for. That's not the greatest idea when you think about what needs to happen for photosynthesis. Another option might be fluorescence, and you can demonstrate this in a lab experiment quite readily. This is where the electron comes down a little bit to a less excited state. And as it's doing that, it will give off a beam of fluorescence light. It's a little bit longer wavelength. You can show that in a simple experiment. That's not what we're going for either. We don't want that situation. Sometimes you'll see energy can get transferred between molecules. So that word, energy, not electrons. I'm not saying that electrons are getting transferred. I'm saying energy. So this is a little interesting to think about. So we're saying that energy transfer can occur, not electron transfer. The best analogy I have for this to get you thinking about this idea that you can wrap your head around is if you've ever been in a music class and they work with tuning forks. And if you've ever seen them strike a tuning fork and it starts to hum, and then you bring another tuning fork next to it, and then that one starts to hum. They're not touching, but there is an energy transfer, and it caused that second tuning fork to hum. And so that is the best example I can give you for what this is. So it's called, the fancy phrase for that is inductive resonance. And I think the tuning fork analogy, the second tuning fork resonating with the first through this energy transfer is the best way to think about it. That will play a role in photosynthesis. So that one is not so bad. You don't really want heat or fluorescence going on, but this inductive resonance, we'll talk about that because that does play a role. That's pretty important. Another thing that could happen is that electron, it gets excited and then it comes down a little bit into what we call a triplet state, which I'm not getting into. That's a lot of physics and chemistry, and we're not spending time on that, but it's a state that persists long enough for that electron to get passed off to an acceptor molecule. That's good. That's what we want. We want for photosynthesis, when we think about that, we want to excite that electron and then get it passed off to an acceptor molecule. And the best way to do that is to get that electron into its so-called triplet state. It's a state of energy where it's stable enough and it sticks there long enough for the transfer of electrons to occur. So that excited triplet state, when they've studied it, it's a little bit more stable than the full-on excitation state. That's very ephemeral and very quick to drop. So it's a little bit more stable. So that means it has a little bit longer lifetime, which means it allows a little bit more time for an actual transfer of electrons. So when we say transfer of electrons, we're really thinking about that's the kickoff to the photochemical reactions, where you actually see this transfer of electrons in an electron transport chain process. Okay, so if that happens, the electron gets excited by light energy, gets to the triplet state, gets passed off, then what we say is that the pigment, the chlorophyll pigment, is now photooxidized, which just means that through light energy, it lost an electron that was given off. It got excited and given off. And then we say whatever molecule got that electron, now that electron, or now that molecule, acceptor molecule, is in its reduced form because it accepted that electron. So we'll talk a little bit more about that idea. Here's a diagram that's trying to get at some of these ideas. So here on the left, you have the ground state of the electrons on the x-axis and then energy on the y-axis. And you can see here that they've hit the chlorophyll with either red light or blue light. And you can see that if you hit chlorophyll, expose them to blue light, you see that it's actually a higher energy state. It goes higher than with red. So it actually does a better job of getting that electron in the chlorophyll pigment nicely excited. And then they're showing here, if that electron comes down a little bit to a less excited state, it gives off heat. Phosphorescence and fluorescence is on here too, but that's what the P and F is, but you don't need to know that. And then here, the T, that state right here on this diagram, see how it's less excited than the initial excitation event, whether it's blue light way up here, red light is a little bit more modest, but both of these you can see if you can get that excited electron to the triplet state, that's a little bit more stable, not quite so excited, and that should allow for that electron to get passed off to an acceptor molecule. If that doesn't happen, then it reverts down to the ground state, and as it's doing that, it gives off a flash of phosphorescence. So, but again, I'm not going to be asking you about phosphorescence or fluorescence. So then if you look at figure B and kind of tilt your head here, they're showing the idea that you have blue light around, 400 nanometers. That's what's getting the electron to the higher excited state compared to red. And then they're showing the absorption peak for red light. And you can see that it does excite the electron, just not as dramatically as the blue. So there's some detail here, some differences in terms of what the red and blue light can do for the plant in terms of excitation energy. Okay. So we're thinking about creating a situation where that electron can get passed from the chlorophyll pigment to an acceptor molecule. So these chlorophylls are what we call photoreceptors. Chlorophyll is not the only photoreceptor. There's plenty of others that a plant has. Plants listen to light or respond to light in many different ways, not just photosynthesis. And so the general term is called photoreceptors. That just means we're talking about pigments that can absorb light. Sometimes that light is used for photosynthesis. Sometimes that light is used for a very different reason. And we'll get into some of the other reasons later, but let's focus in on photosynthesis. So for higher plants, we tend to talk about how angiosperms primarily do photosynthesis and gymnosperms, but we tend to really focus in on angiosperms in particular. So then we talk a lot about chlorophyll A and chlorophyll B. Those are the versions of chlorophyll that higher plants have. And chlorophyll A, well, chlorophyll in general, when you look at it, it has a so-called porphyrin head region, a phytol tail region, and then I showed you a picture before how magnesium is needed to complete chlorophyll structure. And that magnesium is chelated in the center of the porphyrin head region of chlorophyll. So not enough magnesium in the soil, chlorophyll can't get fully synthesized. The plant can't do photosynthesis. You can quickly see how magnesium could be really, really important. So figure A is showing chlorophyll A. So you have the porphyrin head region with magnesium chelated in the center, and then this is the so-called phytol tail that embeds into, so we've zoomed into the chloroplast. That's where we're thinking now in terms of the position in the cell. We've zoomed into the chloroplast and we're looking at the membrane inside the chloroplast. Does anyone remember what that entire membrane is called? Starts with a T, H. Yeah, thank you. Thylakoid. The thylakoid membrane. So we're thinking about if you were to crack open that chloroplast and peer in, you'd see this membrane material just floating around inside the chloroplast. That whole thing is called the thylakoid. That's where all the chlorophyll is embedded. And so you have the chlorophyll, the phytol tail is embedded in the membrane, and then this little porphyrin head region kind of sticks off the thylakoid, kind of dangles. All right, and so this is what we have to imagine absorbing that light energy. And then one of the electrons gets excited and hopefully boosted off, given off to an acceptor molecule. So chlorophyll A is figure A. Chlorophyll B, that's another version of chlorophyll that plants have. And you can see the only difference is you replace the methyl group with an aldehyde group. That's it. And then you can see there's another version here called bacterial chlorophyll A. You can see again, there's more little tweaks there to the structure. This is something that bacteria have. That's why they called it bacterial chlorophyll. So there's some bacteria that do photosynthesis different than what plants do and cyanobacteria do and algae do. They actually have kind of funky photosynthesis compared to what you've been learning if you've learned about photosynthesis. And any other point, K through 12 or in other classes, we tend to really focus in on how plants do it and not how some of these other organisms do their separate version. But anyways, so they have their own pigments. And here again, just showing you the similarity between chlorophyll and hemoglobin. And so the magnesium versus the iron, but you can see the overall structure, the head region is very, very similar. All right, so chlorophyll A, you think about the angiosperms, the gymnosperms, algae, CB stands for cyanobacteria, which are, so cyanobacteria, they used to be called blue-green algae. Do not call them blue-green algae, please, please, please. They're not algae. They're cyanobacteria. So I don't know who nicknamed them blue-green algae, but that's such a bad thing. So there's cyanobacteria. They do photosynthesis very similar to how plants do and algae do it. And they have chlorophyll A. Chlorophyll B, most higher plants have plenty of that as well, and algae have that. And then you have chlorophyll C and chlorophyll D. So again, those versions of chlorophyll, other organisms like diatoms and brown algae, dinoflagellates. Chlorophyll D is really good at absorbing light energy that comes deeper into the oceans, and red algae like to hang out a little deeper in the ocean. And so they tend to have chlorophyll D because that's optimized for the light quality that can make it through the water column to deeper levels. So there's different pigments out there, different photoreceptors, different versions of chlorophyll. And then there's other pigments too that we'll talk about. Like carotenoids. So here are some other pigments. Carotenoids are a group of pigments you're most familiar with. When you eat a carrot, you're eating plenty of carotenoids. And so the orangey yellow colors that you see on plants and, fruits and vegetables, those are carotenoids. And so lots of plants, all plants have that. Protists, many protists do as well. And cyanobacteria. And then phycobilins are another group of pigments better suited to organisms in the ocean. So red algae again, and some cyanobacteria. And then flavonoids are other pigments we've talked about. So far, I think the way we talked about them was it's a chemical that some plants send to the bacteria that fix nitrogen to get them to come to the plant. But flavonoids are also used to color different parts of the plant body, including flowers and fruits, also leaves, some of the beautiful colors we're seeing now in the fall here. The flavonoids are giving that color. So the purpley, pink, scarlet. So flavonoids are a very interesting class of pigments and chemicals. They actually absorb light in the UVB region, ultraviolet B region, which is roughly 280 to 320 nanometers, which that's pretty high frequency. Ultraviolet light can be very damaging. So flavonoids help the plant in other ways besides just being part of absorbing for light energy. So if you were to map out some of these pigments and like hit them with light and see where they maximally absorb, you can see they don't all end up in the same wavelengths. There's all these different peaks in different wavelengths. So, chlorophyll A is the hot pink here, where you can see in the blue, and then in the red, the chlorophyll A. But you can see that the different chlorophylls, they don't do red and blue precisely. There's other quality of light or wavelengths of light that they maximally absorb. And so it's kind of nice that it works out that way for organisms that live in communities, because this allows them, if they have slightly different pigments for photosynthesis, if they're getting shaded out, for example, by another organism, it often doesn't matter because they aren't hogging they're not trying to go for the same light. They can actually use other qualities of light. And so it allows for communities of photosynthetic organisms to hang out together and not compete for the same light source if they're really crowded. So here you're looking at this idea of an action versus an absorption spectrum. Basically, what this is showing, this was one of the first pieces of evidence to demonstrate that photosynthesis is connected to oxygen evolution. So that's why they have it here. They have both absorbance of the chlorophyll pigment, and then they're also measuring the evolution of oxygen being given off. And you can see how perfectly they match up. And so this data was very convincing that through the process of the chlorophyll pigments absorbing light energy and whatever they're doing. Back then, they weren't quite sure what was happening. They just could demonstrate, okay, yes, chlorophyll is absorbing that light energy in the red and blue. And when you see this happening, you also see very high production of oxygen. And so they started piecing together this idea that, this absorption of chlorophyll is allowing for oxygen evolution. So through this process. This is back in like the 1800s when they're trying to figure out what is photosynthesis, how does it work, and what kind of products are made. This is how they were starting to show, oh, oxygen is being made. It matches up so well with the absorption of those two colors. Here's another really simple experiment when they were trying to figure out what's going on with photosynthesis and oxygen. This is a Spirogyra organism that's commonly found in aquatic environments. It's a green algae. It's called Spirogyra because of the way its chloroplasts are arranged. They're in this interesting spiral formation inside their cells. It's very distinct. It's a long organism, very filamentous. So what they did was they took light and hit it along the length of the Spirogyra. And so that's what they're trying to show in figure B here. They took Spirogyra, nice and long, and they hit it with light, all qualities of light. And then they introduced some bacteria into that. They probably had the Spirogyra like in an empty Petri plate in some broth or water. And then they put in some bacteria into that liquid and then shone the light on it. And they were actually, they knew the bacteria needed oxygen to survive. And so they looked for where the bacteria gathered along the length of Spirogyra, and it matched up really quite perfectly to the areas that Spirogyra was getting hit with red and blue light. So super simple experiment, really low tech, but whoops, but again, just showing that, okay, red and blue light seem to be really, really important for maximally, the chlorophyll can maximally absorb. And that's where you get a lot of oxygen production. So that must be really good. Those qualities of light must be really good for photosynthetic processes. So they're trying to piece this all together. And with some very simple experiments, it worked out quite well, with the limitations they had at the time with the technology. They also, started to look really carefully at leaves and the structure of the cells inside the leaf, looking at the chloroplasts inside the leaves. And when you look at a leaf, a typical leaf has a very large surface area. And when you look at a plant in just out in the environment that has to deal with, sunrise, the sun moving across the sky to sunset, they noted that leaves were always trying to orient the blade of their leaf to about 90 degrees to incoming to the sun striking their body. So if the chalkboard or the screen is north, so north, south, west, east, so the sun rises in the east, the leaves would be turning so that the sunlight energy as sunrise is coming would strike 90 degrees. So if my hand is a leaf, it's 90 degrees to the incoming sunlight striking it. That's very efficient for capturing sunlight energy. Then as you go to high noon, the leaves would orient more horizontal to the ground and the sun coming down, beaming down on them. And then sunset, the leaves would turn so they can get that 90 degree again. So they were determining that plants are, that's the best way for them to orient their leaves to really maximize absorption of sunlight energy. And then they started looking at what's happening internally inside the leaf. And they noted that the arrangement of all the cells in there, the palisade cells, the spongy parenchyma cells, all of those are oriented in such a way to give really good optical properties in the sense that when the light is coming into the leaf, it bounces around in there so well that all the cells in the leaf are getting bathed in sunlight energy and no one cell is getting left out. So really good optical properties inside to scatter the light energy around. And then, of course, leaves are highly vascularized so that you can have the products of photosynthesis getting sent to other parts of the body. Lots and lots of chloroplasts in those leaf cells. Anywhere from 20 to 60 is a pretty good average range per cell of chloroplasts. So lots of chloroplasts per cell. And then they also were looking at guard cells that, of course, are guarding the pore of the stomata. And they did notice that a lot of guard cells have lots of chloroplasts, but not every species. So there are some species that have no chloroplasts in their guard cells. But they were gathering all this data, looking at the physical nature of the leaves, optical property of the leaves, then doing these experiments with different qualities of light. And then they zoomed in even further when they had the better technology, of course, to look more carefully at the chloroplast. And so this is old stuff. You've had this idea that it has a double membrane, and then that internal membrane that's just floating around in there is called the thylakoid. Now we need to get a little finer detail. So I'm coming back to this idea. You did learn this already, but I want you to have it in this slide set because I need you to refresh and get these under in your head in terms of when you take that thylakoid membrane and if you were to cut into it and look inside of it, that inside region of the thylakoid, you call it the lumen. And then remember, if the thylakoid is relatively stacked up, then we call that the grana region. If it's relatively unstacked region, we call it the stroma lamellae. All of that though, all the thylakoid contains chlorophyll, okay? Chock full of it. And then that's where we're going to zoom into for the light reactions. And I didn't put on here. I should have put on here. The watery portion inside. The watery portion inside the chloroplast that the thylakoid's floating around in, we call that the stroma. So you need to know that term too, because certain things happen in the stroma, certain things happen in the lumen. And we're going to see that that can be very critical to understanding the process of photosynthesis. Okay, so we're zooming into the chloroplast now for the light reactions. When we think about the second part of photosynthesis, when they make their sugar, that's going to be happening in the watery portion, the stroma. Okay, so we have certain things we're going to find out happen inside the thylakoid in the lumen and certain things in the stroma. So this is an electron microscope picture trying to show pretty well labeled there. It's just that sometimes when they label the thylakoid, they only use one little arrow, but it really should be this. They should have somehow noted that that entire structure is called a thylakoid, all that internal membrane, okay? And then some of it's stacked, so they call it the grana. Some of it's not, so you call it the stroma lamellae. And then the light gray stuff, all of that, that's called the stroma. So you have to imagine, 20 to, 20 to 80 of those per cell in a leaf. So that's, why they always show it as green, because there's all that chlorophyll pigment that's green. Okay, so there's plenty of chlorophyll pigment in the thylakoid membrane. There's also other pigments that are clustering around in certain areas in the thylakoid, and we call them antenna complexes. Very much like, the old-fashioned antenna or rabbit ears on a TV. It's kind of the same idea. These antenna complexes are extra pigments that the plant is using to capture light energy and bring that to an area that's necessary for the photosynthesis process. So they gather light energy and then they transfer that energy to what we refer to as the reaction center, which I abbreviate down to RXN for reaction. All right, so it's very much like, getting the TV signal to your TV. These pigments are capturing slightly different qualities of light, perhaps, and then getting that light energy to the reaction center where you need the actual transfer of electrons to start to happen. So you can see the way I worded it here. It's a transfer of energy to the reaction center, not electrons. All right, so this is where we see that inductive resonance is playing an important role. And so this idea that these pigments are close enough physically where that excitation energy can get transferred without any transfer of an electron. That energy gets brought to the reaction center, and then once in the reaction center, that's when you get actual chemical reactions. So when I say chemical reactions, that's when we start talking about electrons getting transferred. Does that make sense? This is a visual of that, trying to show it. So here they're showing the reaction center, which they've labeled P680. That is 680 is the wavelength of light that it really is good at capturing. Anyways, here you see chlorophyll A, chlorophyll B, carotenoids. So these are all antenna pigments that are surrounding the so-called reaction center, which I'll talk about more. And so they're busy absorbing slightly different qualities of light energy, and then they give that to the reaction center. So figure B is trying to show that idea. So what they're showing with the arrow, 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 that is inductive resonance. It's just that energy transfer to get it all the way to the reaction center. And what they're trying to show here that is that once the reaction center gets that energy, it excites the chlorophyll in the reaction center. So you get that coming out of the ground state energy and getting up higher on the energy system here. So the light energy is actually, being absorbed by several pigments all at once, and then getting that funneled into the reaction center. Here's another way to look at it. So again, they're being very careful here to show that the antenna pigments are doing an energy transfer. So all those green circles or ovals, and they're just trying to show that there's any energy transfer. And then here is a very nice, once you get to the reaction center, you get the transfer of electrons. So this donor is the chlorophyll, sitting in the reaction center. That electron from the chlorophyll gets excited, and then it's given off to an acceptor. So energy transfer versus electron transfer. So they slowly were piecing this together. It actually started back in the 1700s, but it took a long time. This is just to show you that this is definitely a process and very limited by the technology available. So back in the 1700s, they were able to show, okay, yes, oxygen is being evolved by plants through this process of absorbing light energy. And, then Jan Ingenhouse came along and said, yep, definitely light. He did several experiments where he saw no oxygen evolution in the dark, and plants were in the dark versus the light. So very simple, but really helping to piece together the process. And then eventually, a century later, they came up with, okay, we think this is what's happening during photosynthesis. We think the plant is able to take in carbon dioxide through the stomata and reacting it to yield C6H12O6 and oxygen. So when you hear someone say to you, C6H12O6, what do you think of? What's the first thing you think of? What molecule? Glucose. Yep, that's what I think of. Anytime someone spits that phrase to me, I'm like, oh, glucose, which is fine because that's what glucose is. It is C6H12O6. However, glucose is not the first sugar made in photosynthesis. So I think this is a little confusing, especially for an upper level college class. This isn't terribly accurate. So I'd rather you think of it this way. And I'd like you, if I ask you for the equation, please give the second one, where you take a CO2 and react it with water, and then you get CH2O and oxygen. When you put CH2O and then parentheses like that, what you're saying is that's the basic building block of carbohydrate, right? CH2O. It's just the C6H12O6 simplified to the basic building block. That's more appropriate because it's not glucose that's first made by a plant. That's not what they're going for. That's not the sugar. Remember, sucrose is what they make and transport. So that's what they're really going for. So, but anyways, they figured this out and they said, yep, you need carbon dioxide coming into the plant in order to make sugars. You have to have light energy, you have to have pigments to absorb that light energy. And basically what they pieced together is that it's the CO2 being brought in that's being used to build the sugar. So we say it's reduced. CO2 is reduced. That means it's going to get electrons added to it in order to make the carbohydrate. And they figured out that water is the electron donor, or the ultimate electron donor is how they often refer to it. So they have to have a source of water to grab electrons off of, which is incredibly challenging. And CO2 is the acceptor to those electrons ultimately. So this equation is the big picture idea. It is combining both parts of photosynthesis. It's combining the light reactions and the biochemical reactions to make sugar. So it's a very general equation, very big picture equation. It's not really telling you the goal of the light reactions in this equation. When you think about the light reactions from a plant's perspective, this is what they're going for. They're trying to get a good dose of ATP and this other molecule called NADPH, because both of those are heavily needed in order to make their sugars for part 2 of photosynthesis. So that's when you look at the light reactions and look at the whole thing and go, why would a plant do this? That's what they're going for. They want the ATP and the NADPH. Okay. All right. So we're going to continue on with the light reactions for several more sessions, but that's a good start, and we'll get into more of that next time. So moving forward, please bring laptops.